Assalamu alaikum. Our lecture is the viral vaccines. It is for the MSc. Vaccination is a method of giving antigen in order to stimulate the immune response through active immunization. Stimulate the immune response to produce immune elements that will give active immunization. A vaccine is an immunobiological substance that is designed to produce a specific protection against a given disease. A vaccine, though, is antigenic but not pathogenic. We have many immunizing agents. They are either vaccines or immunoglobulins, which is the short-lived, or anti-serum. The first one who discovered the vaccines are Charles Jenner in 1796. He had a brilliant idea about using a cowpox or swinepox virus in order to treat the smallpox disease, which was epidemic or endemic in that time. So, after many trials, smallpox was eliminated as a human disease finally in the 1978. We have many types of vaccines. They are either live attenuated or killed inactivated. Toxoid also are vaccines, another type of vaccines. Polysaccharide and polypeptide, cellular fractions, and the surface antigen or the recombinant vaccines. So what are the live vaccines? Live vaccines are made from live infectious agents without any alteration. The only live vaccinia virus, the only live vaccine is variola or smallpox vaccine is made from live vaccinia, because vaccinia or cowpox, which cause the cowpox, is the virus that cross-react with antigenically, similar antigenic component or composition like variola, and it is not pathogenic to human. So they can use, it can be used, vaccinia virus can be used to give protection and solid immunity to the smallpox vaccine, as smallpox vaccine to the variola. The virulent pathogenic organisms are treated to become attenuated and avirulent but antigenic, retain their immunogenicity. The live attenuated vaccines should not be administered to certain persons with leukemia or lymphoma and other malignancies, because they are immunocompromised, in addition to those receiving corticosteroid and other metabolic agents, those under radiation and the pregnant. The live attenuated vaccines have several advantages and disadvantages. These live attenuated or weakened form of the wild type virus can replicate themselves. So the immune response is more similar to the natural infection and usually effective only with one dose. But severe reactions are possible, especially in the immunocompromised individuals. And we have to worry about creating a wild-type pathogen that can cause the disease in such individuals. However, these vaccines are fragile and must be stored carefully. One important type of vaccine for the treatment of poliovirus infection. We have two types of vaccine, namely Sabin and Salk. The Sabin poliovirus vaccine contains 3 serotypes of the polioviruses in a weakened form, but the live state and is administered orally. They are attenuated by passing these serotypes in a foreign host, monkey kidney cells usually. This is because to select the growth of this virus in another host to make the virus less suited to the original host, which is the human. And subsequently, this virus will grow in the epithelial cells, but not in the nerve cells. that will, which means that it will cause no paralysis. And this type of vaccine will provide local gut immunity, mainly IgA antibody, which will attack the virus in the second exposure to this virus. The Salk vaccine is a polio vaccine that contains 3 serotypes, also like Sabin, but they are inactivated by treating them with formaldehyde and administered by intramuscular injection, but not by the oral. Modern biology have offered or has offered new approaches to make different vaccines. The clone gene from a virus or bacteria, and then this clone gene will express the protein antigen, this protein antigen in a yeast, bacteria, or a mammalian cell in either ordinary culture or tissue culture or cell culture. So this clone gene will taken and then incorporated to another virus, such as the adenovirus, canary virus, vaccinia virus. These viruses are non-pathogenic and they can carry the gene and express the proteins that are produced by these genes, and this live virus and will use as live virus as vaccine. The toxoid, they are prepared by detoxifying the exotoxin of some bacteria, rendering them antigenic but not pathogenic. Actually, adjuvant is used in order to increase, or is needed to increase, the potency of the vaccine. The antibodies produced in the body as a consequence to the toxoid administration will neutralize the toxic moiety of the toxin that is produced during infection, rather than act or attack the organism itself. So they are effective against the toxin, but not the microorganism which will produce the toxin. In general, toxoid are highly effective and safe immunizing. The polysaccharides and polypeptide are cellular fraction vaccines. They have high efficacy and safety. They are prepared from extraction of the cellular fragments. For example, the cell wall polysaccharide antigen, as in meningococcal vaccine, the capsular polysaccharide, which is the highly antigenic and the pathogenic, and the pneumococcal vaccine, and the hepatitis B polypeptide vaccine. The surface antigen or recombinant vaccines are another type of vaccines that are prepared by cloning the hepatitis B surface antigen gene in a yeast cell where it is expressed. Then the hepatitis B surface antigen is used for vaccine. The route of administration of these vaccines are different. We can use the BCG vaccine, for example, in the oral route, or the most vaccine can be taken by the deep subcutaneous or intramuscular route. BCG vaccine can be taken intradermally, while the smallpox vaccine is taken by escartication. Intranasal route is useful for the live attenuated influenza vaccine. The scheme of immunization include the primary and the booster vaccination or immunization. Primary vaccination usually either one dose or multiple doses. The one dose vaccine is given for BCG and variola, measles, mumps, rubella, yellow fever, while the multiple dose vaccine is taken in case of poliovirus, DPT, hepatitis B. The booster vaccination is needed to maintain the immunity level after it declines, after some times has elapsed. The period of maintained immunity due to vaccines differ from one virus to another. In case of cholera vaccine, it is a very short period of time of vaccination, it long for only months. TB vaccine lasts for 2 years, DPT for 3 years, BCG for 5 years, yellow fever 10 years. The solid immunity is given by vaccination with measles, mumps, rubella vaccine, while the level of effectiveness of vaccine either absolutely protective 100% in case of yellow fever vaccine or almost absolutely 99% in case of variola, measles, mumps, rubella, and diphtheria and tetanus vaccine, or highly protective such as in polio virus, PCL, BCG, hepatitis B, and pertussis vaccine, or moderately protective such as in TB, cholera vaccine, and influenza kill